My name is Joseph Hamming. I am a partner in Vital Transformation. I'm a relative newcomer to Vital Transformation, just starting last spring in the April-May timeframe. I want to call the attention to this room before we go on. No one has yet that I remember from today, but we want to thank our friends from Energy and Commerce, House Energy and Commerce, for making this fabulous room available. We've all spoken in numerous places, the basement of the Crown Plaza in Toledo or the Grand Hyatt someplace, and rarely do we have the kind of setting like this. So I think it's very special, and we certainly appreciate everything that they did for us. We're very excited about this meeting, and we're really happy that everyone that has been able to come. We have people that have come from such great distances, and that's one of the things that makes this very special. And this topic is extremely important, better science, better medicine, and everything that goes with it. Right now, we're turning to the area of personalized medicine, and that's particularly interesting given the evolution of personalized medicine today, where it has come in a relatively short period of time. Today we count about, according to the Personalized Medicine Coalition, 130, 140, something like that. Don't hold us to those numbers, of therapies that have a marker, a target, a biomarker, some sort of a test that makes the drug applicable for a particular patient population. When I started my career at Pfizer many years ago, there was a group of intrepid scientists and very brave scientists, I thought, who were trying to gain support from the policy group. I was in the policy group to promote the idea of personalized medicine as something that was coming. This was the early 2000s. There were basically no personalized medicine then. And if you think to 2015, going on 2016, that's an amazing amount of growth in that period of time. Well, if you think about the fact that various large biopharmaceutical firms are boasting 75 to 80% of some of their pipelines as being targeted, personalized medicines, precision medicines, 70 to 80%, that means that in the next 5 or 10 years, this area is going to explode in growth. And I think that's very exciting. It's very exciting for healthcare, but it's also going to create a number of very significant challenges. And those are the challenges that we're going to address today. So like before, you know that the bios are in the booklets, so we're not going to belabor that and read those. But I'd like to introduce Dr. Laura Esserman to start us off, and she's going to start with a clinical perspective and her as an oncologist at the UCSF, the Carol Frank Buck Breast Care Center.
Thank you.
Thank you.
Hello. Good. Great. Perfect.
Very simple.
Great.
Okay.
Perfect. Thanks so much. So I'm here not in my capacity and my academic hat so much as my leader of the I-SPY trials, and I'm here with my colleague Meredith Buxton, who helped me develop the network. And in our reason for wanting to develop it was to advance the science of precision medicine, and it's something that people talk a lot about, but actually putting it into action is sometimes more challenging. And I want to really talk about this as a framework. I'll go through the very specific example of how we've applied this in breast cancer, but there's nothing that's specific about breast cancer, so I'm trying to put it in the context of a framework. So when I think about precision medicine, and I've started building the unit, our multidisciplinary unit at UCSF, and then across the 5 University of California campuses, and then really across the entire I-SPY network, is really thinking about this has always been my mantra, that we need to tailor, and now precision medicine is the term that people use for it, but this is something that, of course, this is what people want. You want their care tailored to the kind of biology that they have, to the person who comes in with their own situation, patient preference, and their own preferences and clinical performance. So how well each of our interventions work in that setting. And the easiest way to explain it to people is to say, well, you know, I could have two people come in the same day who on the surface might look alike, same age, same tumor size, but have extremely different situations, so would have completely different treatments. The efficacy of these treatments completely different. Their access and ability to get health insurance or leave, also very different. Again, genetic predisposition different. And Kim requiring chemotherapy, Veronica needing an excision, perhaps no radiation, and either hormone therapy or a prophylactic mastectomy, depending on her choice. So I actually think that I come from a perspective as a patient advocate. Every week my clinic serves as a patient focus group of people telling me what they think of our treatments, which is often not much, and they'd like something better, what the problems are, and how to fix it. And it's really this perspective that has led me to doing the work that I do today. Because the problem for patients is, you know, and in breast cancer, we have the problem where some people have very indolent disease, and we have a risk of overtreating them. But fully a quarter of all the diagnoses we see are aggressive cancers. Those people are not going to be cured by surgery. And depending on where in the world they live, that depends on what drugs they have access to. I mean, it's amazing the difference. I mean, it took Australia 7 years to get Herceptin in the neoadjuvant setting. India, less than 1% of the population can afford to use it, and they have 30 to 40% of their population with HER2-positive disease. It's pretty shameful, actually, when we look across the world. So I would say that still, in spite of all the progress we're making, people are still dying of their disease. We can do better. It still takes 10 to 15 years, at best, to get drugs to the market. And now we understand it's about in the range of $2.7 billion to get a drug developed. There are lots of things in the pipeline and lots of combinations, but it's very hard to figure out how to test them. There's no pipeline, there's no freeway to try and figure out how to move those drugs along, drop the ones that are successful, let the world know about them so people don't keep making the same mistakes over and over again. And the blockbuster approach, even in immunology, is unlikely to work. And while I think it's true across disease types, we're going to have more uptake of some of these drugs, it's not going to be as good as people think. And 70 to 90% of all drugs fail. So for both patients and drug developers and biopharmaceuticals, it's a problem. No question about it. We have to do better. So part of my way of thinking about the solution is, okay, what's a big part of the problem that we can solve as clinicians and academics? We're a big part of the problem. You know, that when I first started this, everyone would point to the FDA, and I said, well, the FDA is not the problem. We're the problem. We're a problem in the way every trial is like a new discovery or a new, even if it's the exact same drill, we start all over. We start all over because of credit. We start all over because of ownership from one company to another. The list goes on and on. How can you take the white space and start to compress it? What are the things that we can do starting with the patients and the patient treatment? So how do we start with what do people need? How do we identify that person who's most in need of something new and change our clinical process, our clinical trial process, using innovation and learning and adaptive designs or those kinds of things to rapidly change what we do. Create an operational infrastructure that helps you integrate care and research, allows you to really work on real-time data, and then you sort of link up that pre-competitive consortium regulatory guidance and link trial phases, which we believe is actually an important part of diminishing the white space. So that's how I'm going to frame this. So in the specific example of breast cancer, when someone comes in who's got significant disease, node-positive disease, I know they're going to need chemotherapy. I'm a surgeon, right? I understand, much as I would love to think that I'm going to cure this person, I am not. So it turns out the order of therapy doesn't matter. to get the same outcomes, not better one way or the other. But that creates, by starting with the chemotherapy first, I don't take out my biomarker. I can rearrange clinical care to maximize learning and to maximize learning early. And this can be done in almost any clinical scenario, whether you're talking about pulmonary fibrosis or you're talking about colon cancer or myeloma or any number of, antibiotic resistance, any number of things you can think about. How do I change the clinical scenario to re-engineer my care pieces so that I'm maximizing the way in which I get my data? So I can understand what that response therapy is. I can give that patient a better outcome, fewer complications, and I can accelerate drug development to improve outcomes in highest risk women. And I would submit that we shouldn't be testing all these new medicines in the metastatic setting where we have pretty much largely failed to cure people. And maybe there'll be some situations where we can, but probably not. I mean, we can take Gleevec, the poster child for targeted personalized therapy. If you give drug in the blast phase, nobody's really being cured, though we can cure almost 90% of people in the accelerated phase. So I think it's important for us to be thinking about these issues. So in thinking, in trying to imagine what conditions really could enable dramatic improvements in knowledge turns, right? So if you think about what we used to do, we start with the metastatic setting, we do phase 1, then we phase 2, then we do phase 3 in the metastatic setting, then we go to the adjuvant setting, we do surgery, then we give chemotherapy plus or minus another drug, we have to accrue patients, and then we have to do 5-year follow-up. There's just no way, there isn't any possible way you can engineer that down. But if you said, oh, the minute I have a phase 1 drug, I'm going to start with it right now. I'm going to take that 6-month period of time and get that information, and that information can be my driver for accelerated approval, and the follow-up in the same trial could be your information for full approval. That takes real time off the clock, maybe 10 years, just for starters. So it's this idea of taking your clinical scenario, your emerging treatments, trying to identify in whatever your clinical situation is who has high risk for early recurrence. So you have to be able to pick the right patients, and we'll talk about this more as we go on. And then you have to have a collaborative infrastructure and standards for data collection that can further change this. Just to give you a little bit of detail that the trial goes, it is a screening, it's a 2-part trial where we screen everyone gets a molecular profile, and we randomize it. It is a response adaptive randomization trial where we are looking at multiple drugs at the same time to try and figure out which is better. And the trial has multiple arms at once. The trial never closes. It's got a common backbone. We tried to really drive a lot of organizational efficiency and add imaging and biomarker control. So the principles are to test drugs where they matter most, use biomarker and imaging guidance, so if something fails, you at least have a reason why it's not failing, adaptive design, having IT solutions where we're starting to really think about form and function, and getting people to do real-time data collection is a challenge. It's a big challenge. But if we can do that, that of course begins to address the question of how do you get people to collect data in real time and use that to drive all the kind of improvement we want to see down the road and share it. Have that be one of the key pieces of information that can be shared with patients. And to leverage a pre-competitive collaboration model. So this response adaptive randomization could be, we could replace one drug, seamlessly replace it with another, and so on and so forth. So these are the different ways in which we think about it. And part of our purpose was to look for the big signals. We are not the answer of whether the drug has activity. That's not the question we're after. We're after, is there a big enough signal that we think there's a high chance, an 85% predicted likelihood of success in a phase 3 trial, in a relatively small biomarker subset-driven trial. So the subsets are part of the trial. They're not a subset analysis or the different types. So, and then really trying to think about the master IND and testing drugs by class so that hopefully there can be lessons across as much as possible. And really, again, credit is important, democratizing access to data, sharing credit and opportunity, and finding different ways to assign credit. And we've been relatively successful. It's not that we haven't had challenges. You can ask me back. I have a long talk on all the things that I would do differently if I were to start again today, and hopefully we will get a chance to start to do it over multiple times. But we have been able to get many companies to play in the sandbox, and nothing terrible has happened to any of them, I would like to say. We've had 3 drugs graduate, and we were able to really have a big influence on getting accelerated approval moved through the FDA. We've been working with the FDA and the EMA and the PMDA and the TGA to start thinking now in a more global way to think about drug development. And we have now added a phase 1 network and a phase 3 collaborative trial. We've actually screened over 1,600 patients and randomized 900 patients. So the model works, and we definitely can do it. We have multiple organizations participating with this, from the people helping us with operations to all the investigational agents to our device and IT providers. It's been a very interesting learning experience. And the idea of trying to get to accelerated approval, we're not trying to get a lesser standard or some easy route to market. We're really trying to focus on those patients that have the most to gain. So the idea with this was to say, well, if we could, in the I-SPY2 trial, in the phase 2 trial, identify the biomarker pair or the subset of tumors for which that tumor actually works, that we could do a confirmatory phase 3 of that subset, and that with whatever biomarkers that might emerge from the trial, as well as the subset that it graduated in, test it, and then move that on to a confirmatory phase 3, where accelerated approval can be given if you can replicate the pathologic complete response, so the tumor goes away completely, since we know that is associated with better survival, but that you have to enroll all the patients to get to the event-free survival endpoint, and then you can get accelerated approval. But then you can get full approval when that's completed. And we've been working with the EMA and the FDA to do this in concert with the idea that we could move this forward. So I've talked to you about this idea of regulatory guidance being important and how it's really helped us all come to a common pathway. I want to talk a little bit about the linked trial phases. Janet Woodcock in particular had pushed us to really move forward with the with this idea of I-SPY3, and I couldn't quite understand why that was important at the time, since I had nothing better to do than to think about start another network. But I think her idea is that there's a lot of white space, right? If time is the key, and every manufacturer, every drug developer knows that time is the key. So for us, if you think about how the clinical care system works, you've got to get the engine running. If you can get lots of people and lots of centers where, someone comes in, instead of 5% of my patients getting neoadjuvant therapy, every person that needs neoadjuvant therapy is getting it. So of the 25 to 30% of breast cancers that are serious, everyone's getting neoadjuvant therapy. And if 50% of those patients go on trial, okay, you could get all this, it would take you a year at most to get these drugs through. And then when you have those drugs approved, you can start figuring out how to optimize them in clinical practice. So I think that's and to build automation and analytics in the trial, there's tons of ways in which you can continue to drive out that white space. So, and so one of the things that we realized is we would tell people, okay, here's our backbone. We figured, that people would just go get the phase 1b data, because you have to have safety data, because patients with breast cancer, actually their standard of care happens to be chemotherapy. So whatever your standard of care is, you have to have that plus your new agent for safety data. But people would languish, they wouldn't have it, it could take 3 years, you'd be surprised. So we've started within our network. We have 7 centers that are participating in our phase 1 network, and we're working on now a phase 3 international confirmatory trial. And the idea is, you've got the engine running, the minute you know you've got your dose, you write your amendment, and it's a month to get it into the trial. And the minute you know something's graduating, you've got your engine running, even if it's people commonly doing it, you're dropping it into the trial, boom, you could really make incredible progress. And so we're starting to build out the I-SPY3 footprint. We're working on getting our first drug in. We actually have the protocol, actually has already been reviewed by the FDA and the EMA, and it is again a master trial standing platform. We have shared control groups. We have a data plan that's submitted as part of the master trial. Again, focus on efficiency and collection of data elements that directly influence your primary endpoint, and the rest, we don't have to be is not so important. We were actually strongly influenced by Rob Califf, who's talking tomorrow, and hopefully will be the FDA commissioner. But that's sort of what he did in the Gusto trials in the cardiology field, and he's absolutely right. And this whole idea of where does this data come from that you need? The stuff that can come from patients should come from patients. The stuff that should come from physicians should come as a checklist and should flow through as data liquidity. Having it be open label and not double blind. You can't have a master trial if it's double blind. So what do you need to do? You need to understand what your endpoint is, which is pathologic complete response. You get your pathologist to do it on a handheld device, not knowing your trial, not knowing who's on trial. And that's actually been our solution. So there's lots of ways to think about it, but it's something that could increase the capacity around the globe to work effectively to get drugs through, an opportunity for regulatory harmonization, and a way of advancing the concept of integrating care and research. I think it will promote the adoption of standards. Earlier this morning about having data that's interoperable. There's no reason. The checklist is pretty clear of what you need. That's what you need to take good care of somebody. It's not some mystery. And if you have these centers of excellence, you can have lower costs of trial. We don't need 200 sites open with one patient. What you need is centers that are going to take care of patients like that and put it into practice. And the people who are sick, send those people to the specialty centers where they can get that care, or at least have it coordinated by those care so that you have better access issues. And at the end of the trial, every site we touch would be better than they were before. So this is this idea of having e-source or electronic source, this idea of one source of data collection. And what we mean by this, and I just put this in because this idea, if you enter the data right once, you could actually have it used for any number. You can have, if you get the key data elements, you can have registries, clinical trials, trial matching, quality improvement. That, to me, is what we really need to make personalized medicine go forward.
Now, keeping the same theme, we're going to go across the pond and get the perspective from Richard Barker from Catapult. He's going to tell us about its Catapult from CASME. So, thank you very much.
I have two roles, Joe, yes, as director of CASME and also as chairman of the Precision Medicine Catapult. And thank you very much, Dwayne, Joe, and our sponsors for enabling us to be here. It's great to be back in this room. I was last in it about 20 years ago. It hasn't changed. It's just as fantastic. So if you don't want to listen to me, have a look at the ceiling. It's fantastic. But I'd like to just underscore some of the things that have come out today by telling a very brief story. In Alaska, in Anchorage, Alaska, there is the Lundfeldt family. And a few years ago, they had their first child. They christened him Cameron. And Alyssa Lundfeldt was obviously very proud right at the beginning. But unfortunately, the child didn't thrive, didn't meet its development goals, or his development goals, and started to turn gray, eyes rolled back in the head, listless, and so on. Took the child to first the emergency room and lots of other specialists. Ultimately, Cameron was diagnosed as the youngest patient with diabetes, type 1 diabetes, in the whole of Alaska. So what's happening now? Is he injecting insulin 3 times a day, watching his food? No, because someone in the mix said, hey, juvenile diabetes at that kind of stage may well have a genetic component. And so there was a connection made between Anchorage, Alaska and University of Chicago, where they had been doing some leading work, and also with the University of Exeter in the UK, where they'd also been identifying a particular genetic abnormality called KCNJ11. And the child was tested for this, and turned out that was the variant that his genome had. The interesting thing here, and there may be a diabetologist in the audience, in which case I should shut up, but the child's pancreas makes plenty of insulin. The beta cells are full of insulin, it just can't get out. The transport mechanism doesn't work. And all you need to do is give a simple oral drug, a sulfonylurea, and that insulin comes out and the child is normal. So instead of injections several times a day, and you can imagine all the healthcare costs and time and agony associated with that for the rest of his life, he just takes a simple oral pill. And that seems to me to be a story that has got a number of elements to it. I mean, the first thing it does is really reinforce the point that we are entering an era of precision medicine based on those kinds of molecular insights that fundamentally transforms the way we define disease. And I think Hans-Georg made that point earlier today, but I just want to underline it. We still talk about the diseases defined by symptoms. In fact, in practice, we're going to be defining them molecularly. Goodness knows what medical training is going to look like then. We probably have to reword almost all of our slides because we're talking about very specific molecular defects with very specific therapies. It also brings together pharmaceuticals, diagnostics, and informatics in ways that have never been converged before. Amy made these points tremendously powerfully. 3 different industries, or there's probably more than 3 different industries, have to come together to make this work. It's also a movement able to bring interventions much earlier in our lives, not perhaps just when disease symptoms appear, as they did in the case of Cameron, but you've all probably read about Human Longevity and Arivale. These are companies being set up to actually sequence people right at the beginning of their lives or as soon as they go on the program, anticipate the disease, and take preventive measures that mean the disease never actually appears. So personalized or precision medicine becomes precision prevention in time. But the other thing that the Cameron story reminds me of is that these insights are actually able to re-engineer care pathways and workflows in quite fundamental ways. And that's really important. And in a minute, we're going to listen to Mark, who's going to tell us the solution to the economic problem. But I think part of the solution to the economic problem is to recognize those places where these technologies fundamentally simplify and rationalize the processes of care, not just from the point of view of the patient, but from the point of view of the workflow within the hospital and throughout. And therefore, I think precision medicine is crucial to making healthcare itself sustainable. About 4 or 5 years ago, I wrote a book that said avoiding a medical meltdown, because I think we see that in so many of the healthcare systems of the world. Certainly in the UK, it's running out of money really fast. And what we have to do is not present precision medicine to these kinds of folks as extra expense, but as a way of revolutionizing the way in which care is delivered and actually making it fundamentally sustainable. But I think a number of other things that the Cameron story doesn't reinforce, but have come up today. Firstly, that precision medicine is central to this idea of adaptive development, adaptive pathways, accelerated pathways. Without that ability to target, we can't do some of this adaptive thinking, adaptive mindset that Hans-Georg and others have talked about, and Sabine really elegantly laid out. It enables, indeed, I believe it demands what Laura was telling us about new clinical trial designs. Without precision medicine, we can't do those trial designs. They don't make sense. Now they do. And of course, it makes sense, as Ken was telling us, of all this real-world data, if we can just ensure its reliability. The Precision Medicine Catapult, by the way, is a UK initiative to grow the industry of precision medicine. Now, some of you may react against, as I initially did, this idea of an industry. But when you add up pharmaceuticals, diagnostics, informatics, data analytics, this is actually an emerging industry. And what we want to do is grow it in the UK, but to do it with international collaborations. Laura and I have already tried to address the fact that I didn't see the UK shaded in on your charts. So we need, the French were and the Germans were. So obviously we need to get your travel itinerary worked out. Because we have in fact done the 100,000 Genome Project. And breast cancer is one of the key elements of the 100,000 Genome Project. So there'll be thousands of patients for which we have the whole genome sequence and can put them in. I think what I'd like to do in just the last few seconds is to throw the challenge back to Mark that he's going to address, right? I knew you'd needed that. The economic value question, I think, is central to this. And Amy made the point extremely powerfully. You know, if you're a diagnostic lab director and somebody comes along and says, how would you like to spend another few hundred or few $1,000s on this test? Of course you don't. Somehow or other, you have to find the person who has a view of the total care pathway and says, actually, if you use this test, even if it's $10,000, it might save $50,000 in useless therapeutics. So what we have to do is get the economic models moving as fast as the science, which I'm sure is something that Mark is absolutely equal to. with regard to the therapeutics value, the diagnostics value, and the value of the changes in the care pathway that we need to see. So pricing and payment, I think, will increasingly be the themes that precision medicine has to tackle. Thanks very much.
Really great. That was terrific. Thank you, Richard. So as we've just been promised, Mark is going to come up and explain how all of this is going to get fixed and how we're going to pay for all of it. Mark is the executive director at the executive in residence, excuse me, at MIT.
I never leave, I always sleep. And Richard, thank you. You said you would set me up for the final talk of the day, and I think you set me up royally. So thank you very much for all of that. I'm your last speaker for the day, and I have the job of not only addressing some of the issues, but making you love economics along the way, since that's my role here today. So before we think about the value, the economic value of stratified, personalized, precision-targeted, or otherwise named medicines, I thought we'd think for just a moment, very briefly, about what did we think of success in the empirical, the old days, right? And one of the best examples that we often tout is how the cardiovascular disease rates have fallen over the past 10, 20, 30 years. And this has been a fantastic success as we think about this. And as you look at that, statins, while only part of the solution for that, played an important role. They were empirically prescribed to those who display certain risk factors. And the real objective was to avoid one fatal event by giving the drug to persons on average for 5 years. You could expect to avoid an event, but not for the one person. You had to give it to 83 people, right? We just didn't know which of the 83 was going to avoid the fatal heart attack. All right? So if you treated 83 people for 5 years, right, and the cost or price of the drug